The next member of the girl society is lysosome. Lysosome is one of the very important cell organelle in eukaryotic system, which is assisting in the synthesis of lot of hydrolyzing enzymes. As these lysosomes are of two types, they are either primary or maybe secondary in nature. The secondary lysosomes are of three types. They may be autophagosomes or they may be heterophagosomes or they may be residual body, or you may call them as telolysosome. So they may be also called as a part of lysosome, telolysosome. Maybe one of the very interesting name we can use for it. About 50 types of hydrolyzing enzymes have been recorded from different types of lysosome. Some of the very interesting ones are, say for instance, some of them may be hyaluronidase. Some of them may be acid phosphatase. Some of them may be beta galactosidase, etc. As they are associated mostly with the digestion of cell components, so the functional review may be of two types. You can summarize them under extracellular and intracellular digestion of cell components. A very pertinent point is the lysosome, for this excellent capacity of cell digestion, is now called as suicide bag. We are now going to the next very interesting concept of next cell organelle. The important most cell organelle now we are dealing with is an excellent cell organelle exclusively found in animals, and this is centrosome. Each centrosome is called rather diplosome. Why? Because it consists of two centrioles. I am clarifying this situation with a clearly specific diagram. Let's see the diagram and we can identify how we are saying this is a centriolar microtubular network. Each centrosome, they are containing two barrel-shaped centrioles, mostly at right angles to each other, supported by centriolar microtubules, which are supported as astral rays. So a number of astral microfilaments are surrounding paired centrioles, constituting the beautiful language diplosomes. Actually, you mean to say these are centrioles and they are astral microtubules. centrioles. The centriole, or better to say, the centrosome. It is a unique structure in eukaryotic animal systems because centrioles are assisting in flagellary propulsion, sperm movement, and formation of spindle fiber during the act of cell division. Now, a very important thing is, it may be a very interesting question that can you name a plant cell possessing centrosome? The answer is Chlamydomonas, which is an excellent example of plant possessing centrosome. That is an excellent exception also. If I make a drawing of the centriolar microtubular structure, it is supposed to be having a design of 9+0 microtubular arrangement. If you ask me, sir, why it is a 9+0? So let me make a very specific and schematic diagram. Here we are getting individual 9 rods, 3, 4, 5, 6, 7, 8 and 9. Here it's a central oriented structure and each filament is divisible in 3 subunits. If I make a drawing like this, they are supposed to be with 3 subunits each, A, B and C. And they are connected with each other with this type of CA connective. So if I make a drawing like this, they will be connected like this. So I think they are the 3 tubules actually. They are oriented in this fashion, A, B, C. The next you are getting again A, B, C and A, B, C, they remain connected with CA connective. So actually this is the centralmost design. Here this is called hub. So the whole structure is actually 9 individual microtubular rods. And as we are specifying 9 individual microtubular rods, no individual other rod is involved, so that's why it is 9+0 microtubular arrangement. So the next very important cell organelle is coming automatically is plastid. Now if you say plastid, plastid is a very important cell organelle exclusively in eukaryotic plant cells. So this is plastid. So if you ask me how many types of plastid you are getting, that the name goes to 3 types. This is leucoplastid, this is chloroplastid, and the last one is chromoplastid. So the leuco is colorless, responsible mostly for the storage of different emergency food. Chloroplastid is having a green color, which is making the chlorophyllous structure for photosynthesis to continue in leaf. The chromoplastids are in different colored shades other than green, obviously. So if we clarify how many types of leucoplastids we are getting, the very important structure is leucoplastids may be of 3 types, as they are mostly for storage of food. So how many ways you can classify them? So this is amyloplast, obviously for sugars. The name goes to proteinoplast, obviously for proteins. The next is elaioplast, obviously for fats. Different types of chromoplasts can also be tabulated as xanthoplast for rendering a definite yellow color, carotenoplast for orange, pheoplast, say, for brown colors. So now we are going for the composition and design of an ideal chloroplastid molecule which is mostly abundant in leafy photosynthetic areas of higher plants. If I make a drawing like this, a bimembranal or double membrane bound structure, rather this can be stated like this with a definite two membranes, outer membrane, inner membrane, and this is a very important zone. We call it periplastidial space, I must say. So here we are getting small sac-like structures in circle type of patches. A lot of such type of structures you are getting, which are sac-like structures containing about 250 chlorophyll pack. Each pack is called quantasome. So these quantasomes are present in these structures which are supposed to be thylakoids. Right. So this is the stroma and this is the grana, where effective photosynthesis is expected to continue in chloroplastid of higher plant cells. But it's a very important and conceptual area also. Prokaryotes are lacking plastid. I mean, prokaryotes are lacking every cell organelle excepting ribosome. So, if prokaryotes are allowed to perform photosynthesis, questions arise where they will do it. They will do it in chromatophore or in chlorosome, especially the prokaryotic means the photosynthetic bacteria. Now, after a lot of discussion with a number of photosynthetic components, chloroplastid, chromoplastid, leucoplastid, regarding storage of food, making different colors to the floral parts, assisting in pollination and in photosynthesis, obviously. Now, the greatest is also it, which is an exclusive member of the cell organelles distributed mostly in eukaryotes as well as in prokaryotes. So if I ask you, can you name one cell organelle which is present both in eukaryote and prokaryote? The answer goes to ribosome. So they may be of two types, 70S and 80S. 70S for prokaryote, 80S for eukaryote. Very interesting, in your books you'll be getting, the 70S is dimensionally differentiated into 50S and 30S. The bigger one 50, the smaller one 30. In eukaryotic system, the bigger one is 60 and the smaller one is 40. So they are the two individual sub-units, better you can say. So one can ask you, what is S? According to the concept of Wedberg, this is the sedimentation coefficient as specified by Wedberg. So ribosomes are actually classified on the basis of Wedberg's sedimentation coefficient. That is a very interesting one. Ribosomes are called protein factory. If you make the ribosomes, they are the protein factories. And ribosomes, why they are called protein factory? Because they are mostly assisting in the protein synthesis. It has been reported by Spirin that in addition to ribosomes, in eukaryotic higher cells, some specific components are present which are called as informosomes. Informosomes are free mRNA molecules ready to carry biological informations. So they are a specific type of eukaryotic cell component as recorded by Spirin. A very interesting information is about the different types of cell organelles that mitochondria and chloroplast both are called as semi-autonomous cell organelle. So a very important thing is why they are called so. Because they have three unique points to understand. Number one, both of them contain 4 to 6 strands of free DNA. Number two, both are having their own ribosomes for independent protein synthesis. Number three, the last one, both are originated independently from their individual precursors. Thank you. For testing your understanding of this lesson and more videos, log on to www.tubelessons.net.